Greetings everyone, my name is Thabile Madlala and I'll be doing a presentation of vaccine design against Toxoplasma gondii using surface antigen 1 and dense granular protein 6 antigens through immunoinformatic techniques. Toxoplasmosis is a zoonotic disease caused by an intracellular obligate parasite called Toxoplasma gondii, which often results in congenital infections and abortion in humans and livestock. This parasite is normally found in cats, which serves as a primary carrier or host for the parasites, and other warm-blooded animals, which serves as intermediate hosts. The parasite threatens public health and animal welfare due to increased zoonotic risk of transmission to humans, as it is transmitted through consumption of contaminated food, being in contact with contaminated water or vegetables. In human, it is often transmitted through ingestion of poorly cooked meat containing the parasite tissue cyst or being in contact with contaminated water or vegetables. And the aim of the study was to explore the Toxoplasma gondii antigens through identification of antigenic T cell and B cell epitopes and to design a potentially cost-effective vaccine through immunoinformatic techniques. Now, this was achieved through a series of computational steps involving the retrieval of amino acid sequence of sheep Toxoplasma gondii surface antigen 1 and dense granular protein 6 from NCBI. Now, the retrieved sequences were then subjected to multiple sequence alignments in cluster W and structural and transmembrane structural property analysis to generate conserved sequences. The conserved sequences were then further subjected to an online server called Immunoinformatic database and analysis resource in order to generate or predict our T cell epitopes, which are your CD8 and CD4. Now, to confirm whether the generated epitopes were in fact antigenic, 100% conserved, and non-allergenic, they were subjected or assessed for antigenicity, immunogenicity, allergenicity, and conservancy. The CD4 epitopes that were identified or predicted were further assessed for interferon gamma and interleukin-4 inducing properties, where all the predicted or identified epitopes which fall under the set criteria in all these analyses were then regarded as our final T cell and B cell epitopes. Okay. Now the final predicted epitope were then joined together to form our multi-epitope vaccine construct, which was further subjected through a series of validation steps. Okay. Now we were able to identify 2 T cell epitopes and 10 cell epitopes, of which they were joined together using GPGP and KK linkers. A cholera toxin B subunit was also added to our vaccine as an adjuvant in order to enhance the immunogenicity of our vaccine. Now, the tertiary structure of our vaccine was predicted using an online server called 3D Pro, which was further refined using Galaxy Web Server. Now, the tertiary structure was validated, or the quality of our design tertiary structure was validated using a PROSA, where z-score of -2.93 was observed, indicating an overall good quality of our designed vaccine. Okay. Now, the designed vaccine was further developed using through the CHPC platforms with toll-like receptor 4, where out of the 50 confirmations that were obtained, the model just selected had the lowest binding affinity of -151.645 kilocal per moles. Now, physiochemical properties of our vaccine was further assessed, where the properties were observed, or where it revealed that our vaccine had a molecular weight of 34.08 kilodaltons, and it was observed to be highly antigenic with a score of 0.9135 and soluble with a score of 0.9485. It also was observed to have theoretic RPI of 9.81 and an instability index of 29.35, indicating our vaccine to have a protein which was basic and stable in nature. The vaccine was also observed to have an aliphatic index of 75.62 and a gravy of minus 0.425, indicating that our vaccine was further thermostable and hydrophilic in nature. The designed docked structure is seen in Figure 2. Okay. Now, with the current study, we're able to design a promising multi-epitope vaccine using a cocktail of the T gondii antigens mentioned before. The designed vaccine was observed to be highly antigenic and non-allergenic, which is promising for the production of vaccines in the future. Yeah, moving forward in the study, we still have to compare the structural stability of our vaccine in relation to other toll-like receptor, which is TLR4 and against TLR2, in order to assess whether our vaccine has the potential to trigger the immune responses when administered into a host through an online immune simulation server. But these findings serve as a valuable source of information for future studies focusing on vaccine development against T. gondii. Thank you.